So our next speaker today is Dr. Anuradha Budha. I'm excited because this summer we're making the most out of Zoom. And so typically we only have UBRP faculty who give seminars, but hey, since we're on Zoom, we're inviting people from all over the place, and we're making an effort to bring in alumni as well. So Dr. Budha is currently working at the National Institutes of Health. She's a senior associate scientist and a program manager for the National Cancer Institute's Center for Cancer Research's, Center for Cancer Research's Liver Cancer Program. And I'm sure she will explain more. Dr. Budha was in UBRP from 1993 to 1995, where she studied genetic expression patterns in the larval brain of Drosophila, speaking of insects. And she also participated in Bravo, which was our international arm of UBRP, and she was able to go continue her research at University of Adelaide in Australia. Dr. Budha completed her bachelor's degree in molecular and cellular biology and biochem, and then she went to Cornell and finished her PhD. And then in 2002, she joined the National Cancer Institute as a postdoc, and then she was promoted to staff scientist and then to associate scientist. And now, as I mentioned, she's a senior associate scientist. Dr. Budha's work focuses on the genome scale analysis of human liver cancer. She's authored more than 50 publications and 6 book chapters, and she has many awards, including the Center for Cancer Research's Outstanding Postdoc Award, the National Cancer Institute's Directors Innovation Award, and the National Cancer Institute's Directors Award. So thank you so much for being with us today. The floor is yours.
Thank you so much, Jennifer. It's a pleasure to be with all of you. It's nice to see all the faces of the young scientists. Okay, so thanks again, Jennifer. It's a real pleasure to be here today and to share a little bit of our science with you. So Jennifer went through most of this. She asked us to sort of give a brief synopsis of our scientific journey. So she mentioned, I kind of went from fly brains to human cancer. So that's a bit of a leap. So I started at the University of Arizona for a bachelor's degree on the biochem department, and was participating in UBRP and Bravo through Scott Selleck's lab, who had joined that summer that I started. And so I ended up in a laboratory with just boxes and empty benches. So it was kind of nice to see a lab literally start from the very beginning and to be a novice in the laboratory, having never done research before. So it was quite an interesting beginning. So there I studied central nervous system development in the larval brain. So a lot of microscopy and staining procedures. Then I went to Cornell for my PhD and went into a completely different area, studied retinoid biology. Started with a biochemistry project focusing on ligand binding dissociation rates and measuring those in receptors that were associated with cancer. And that led to a more specific project on mammary cancer. And that's where I learned a bit about cell lines and molecular biology. And then from there, I joined the National Cancer Institute as a postdoctoral fellow in 2002 in the Laboratory of Human Carcinogenesis, which I am still a part of. And there I began studies of human liver cancer, mainly studying hepatitis C virus, starting with the very early NCI-made gene arrays, which are very archaic now compared to all the fancy sequencing that we now have, and some studies on small RNAs, the microRNAs. I then became a staff scientist and studied functional and integrated genomics in liver cancer. And this led me in the direction of global studies of liver cancer, where I had to learn a lot of epidemiology, which I certainly hadn't taken any coursework on prior. So this was, again, another sort of leap into a realm that I hadn't been in prior, and into bioinformatics because of all the big data I was dealing with at the time. And then as Jennifer mentioned, in 2018, the NCI initiated a liver cancer program to focus in on this particular cancer, and I became the program manager there. So since been leading programmatic efforts focused on liver cancer across the US and the world. And so that's a bit about what I'm going to tell you about today, is some of the studies that I've been involved in dealing with our understanding of human liver cancer. So liver cancer is a leading cause of cancer death worldwide. About 90% of liver cancer is hepatocellular carcinoma, which I'll refer to as HCC. and it mainly occurs in Asia and regions in sub-Saharan Africa. The majority of HCC is attributable to infection with hepatitis viruses, either hepatitis B or hepatitis C typically. And patients usually have a very poor outcome with this disease. And this is generally due to late disease stage presentation, heterogeneity of the disease, and there's currently lack of effective treatment for liver cancer. And so we need to understand a lot about the underlying biology of this disease due to a variety of different factors, and to improve our methods for diagnosing liver cancer, predicting its outcome, and to try to identify novel therapeutic options to improve patient outcome. The liver cancer worldwide, I mentioned it's most predominant in Asia. There's gender disparity, approximately about a 3-to-1 ratio, where males are more prone to developing liver cancer than females. And if we look at the incidence versus mortality rate for liver cancer, it's certainly not what we want to see. Essentially, the rate of incidence is equivalent to the rate of mortality. And so this is a very deadly cancer. In the United States, there are about 42,000 estimated cases of liver cancer in 2020 for both sexes, and approximately 30,000 deaths. And it's in the top 10 leading causes of death in both males and females in the US. Incidence rates of liver cancer are rising in the United States, and this is atypical of many other cancers. It's one of sort of the oddball cancers that's rising. We can see that here just in state-specific age-adjusted HCC incidence rates from 2003 to 2012. And we can see here that the color represents a greater incidence rate. And we can see quite a dramatic increase across that decade or so. And the incidence rate is increasing in certain populations, Hispanic and Black populations, when compared to white and Asian. In terms of mortality, here's the age standardized mortality rate as of 2014, the latest data that we have. And we can see here that there are certain hotspots of liver cancer within the United States, predominantly in southern regions, with higher levels in the southern regions of Texas and parts of Alaska. The bottom graph here shows the percent change in age standardized mortality rate over a 2-decade period. And we can see that this percentage here corresponds to a percentage, between the 135 to 173%. So this is quite a huge percent change in the mortality rate for this particular cancer. And it's one of the top cancers in an NCI study among men and women. So as I mentioned, liver cancer is a very heterogeneous disease. There are many different factors that are related to a person's their tendency to develop liver cancer, including their lifestyle, environmental factors, demographics, as well as a variety occurring within the tumor itself. And so these different causative factors can elicit distinct molecular mechanisms that lead to transformation of the cell. And this results in this vast molecular heterogeneity that we observe among patients. So how do we deal with this heterogeneous disease? So our lab and others are using this particular methodology of building a global liver cancer data ecosystem, where we integrate biobank and information commons to help guide interventions based on the identification of biomarkers and signatures. And so our lab and others have been involved in case control studies and developing cohorts where we collect patient specimens, and we perform various downstream omics studies like gene expression, metabolomics, microbiome assessments on an individual level, as well as an integrated way, in concert with clinical and histopathological classifications. And our hope in doing so is that we can find more homogeneous groups of individuals based on their molecular signaling patterns, which will help us to improve target identification, diagnosis, prognosis, treatment, et cetera, with the hope of really impacting quality of life and survival by using these biomarker-guided interventions. So I'll give you, in the next few minutes, just 3 examples of how we've used this methodology to answer certain questions on liver cancer. So most of these questions are dealing with trying to identify molecular subgroups, so these more homogeneous subgroups of patients. And in this particular case, looking at it in relation to race and ethnicity. We've also looked at it in relation to viral exposure, as well as to particular treatment regimens. And we've used a variety of different studies that we've initiated in order to answer these questions, or at least begin to answer these questions. And so the first sort of snippet that I'll go through is a study that we've conducted in Thailand. And why Thailand? So in Thailand, liver cancer is a primary cause of cancer-related death. It's a major health burden in that country. And I mentioned that most of the primary liver cancer that occurs worldwide is HCC. However, in Thailand, a second form of liver cancer is quite prevalent called cholangiocarcinoma, or CCA. And CCA occurs very predominantly, mainly in the northeast region of Thailand. You can see here CCA is the sort of red colored bar, and we can see here just the predominance of it in this region of Thailand compared to other countries and other regions of the country. And while hepatitis viruses are major etiological factors for hepatocellular carcinoma, it's thought that an infection with a liver fluke is a major etiological factor in cholangiocarcinoma. And in certain populations, particularly in the Northeast, there's a consumption of raw fish that's infected with the liver fluke, and it's been associated with a higher level of CCA in that region. It's a very understudied area in liver cancer and liver biology. And so in 2009, the NCI initiated a consortium with Thailand, which we call Tiger LC. It stands for the Thailand Initiative in Genomics and Expression Research in Liver Cancer. And it's a case control study where we're enrolling 6,000 individuals with hepatocellular carcinoma, with cholangiocarcinoma, or those at high risk for developing these two cancer types. So either those with this liver fluke or infection, or those with chronic liver disease, like hepatitis infection or fatty liver disease. And we have matched healthy population controls. And the aim is to use both biospecimens collected in this cohort and questionnaire data to define clinically relevant biomarkers for risk assessment, for early detection, for tumor classification, prognosis, and response to therapy. And the NCI is working with several universities and hospitals across Thailand in order to conduct this study. And it's being conducted within Thailand and coordinated by the Chulalongkorn Research Institute, which is headed by Her Royal Highness, the Princess of Thailand, who happens to have a PhD and is quite interested in liver cancer research. So here's just an example of some of the data coming out of this study. This was our phase 1 study from Tiger LC. It was published in 2017, where we looked at the first 150 or so consecutively enrolled patients, looking at their tumor tissue. And we conducted a transcriptome study looking at their gene expression, an exome study looking at their mutation status, copy number variation status, as well as their metabolome. So what biochemicals are present in their tissue and in their serum. And the questions we're asking again is, are there particular molecular subtypes at each of these data levels that we can identify? And we also integrated the data across these different platforms, and then validated the data in additional cohorts that were available across the world. And so I'm not going to go into the specific data, but just to sort of schematically show that integration of the omics data allowed us to identify two major subgroups that were of interest, C1 and C2. And these subgroups were associated with patient outcomes. So the C1 patients, that were the patients that had a molecular signaling that would put them into the C1 group, had a very poor outcome when compared to patients in the C2 group. And we found that a specific particular molecular pattern that was present in Asian patients when we compared this cohort to other cohorts around the world. And this particular molecular signaling was related to an alteration of metabolic processes, specifically bile acid metabolism. And we also saw differences in the liver microenvironment with alterations of particular T cell populations. And in concert with the tissue data, we also identified metabolites that were expressed in patient serum that could also distinguish these two subgroups. And so this now allows us to potentially utilize this data for biomarker-guided interventions within this population. So Tiger LC is an ongoing study, and we have many future studies and current studies lined up to go. As you can see, many of them are omics-based, and we also have sort of an epidemiological portion of the study as well, to try to understand what are the root causes and risk factors leading to liver cancer in that region of the world. Now, another area of the world that we have started working with is Mongolia. And Mongolia actually has the highest level of liver cancer in the world. Even among other Asian countries, its rates are tremendously high. It's about 4 times higher than other Asian countries where HCC is prevalent. They have a very low 5-year survival rate, below 15%, and a very interesting underlying etiology where they have a double infection of the virus. So hepatitis B that we typically see in regions of the world, but they also have a co-infection with hepatitis D virus, which is a very understudied hepatitis virus family. And so this is a quite interesting virus to be studying. And there may be other etiological factors that are associated with the risk in this population that we haven't really understood at this stage because it's such an understudied population. So the NCI has joined with the Center for Global Health at NIH, as well as the National Cancer Center in Mongolia, to conduct a study, a case control study in Mongolia. And we recently published the first global transcriptome study in this population last year, where we explored the tissue, both liver tissue as well as liver tumor tissue, as well as corresponding non-tumor tissue from 76 Mongolian patients with liver cancer, looking at their gene expression level, as well as their mutation panel data. And what I'm showing on the center panel here is just a heat map of the molecular profile of these patients. And we can see here that we've been able to subgroup these patients into 4 molecular subgroups. And 2 of the subgroups, the ones on the left, if we look at the Kaplan-Meier curve on the bottom here, they have a much better survival than the remaining 2. And so the molecular subgroups are related to patient outcome. And so we can then compare the Mongolian data with other data sets across the world, like TCGA, which is a large data set within the United States, our Thailand cohort, et cetera. And what we can see is that for certain subgroups, for example, this M1 subgroup that's related to good survival, we can see that it's quite similar across different populations in the world that have been studied, whereas other subgroups are quite different. And so that warrants further exploration. So for our Mongolia study, we're able to identify 4 outcome-related molecular subgroups. They show some similarity and some differences among other cohorts that have HCC. And what I didn't show you was the mutation profiles among the patients, where we were able to identify certain driver genes that are associated with the 2 subgroups associated with either poor or good outcome. So moving on to another question related to molecular subgroups. In this case, are molecular subgroups related to patterns of viral exposure? So we're all interested in viruses, especially after this year. And we've conducted a study with the University of Maryland in Baltimore to begin to address this question. And so we started the study back in 2009. It's a case control study in Baltimore, looking at patients with primary liver cancer, hepatocellular carcinoma, and patients who are at risk for developing the disease. So these are patients with chronic liver disease, and we're following them across time to see who develops cancer and who doesn't, and healthy match population controls in the region, with an enrollment goal of 2,000 individuals, with biospecimen collection, and again, a questionnaire. In this study, we used a method called VIR-SCAN, which was developed by Steve Elledge's group at Harvard. And essentially, it's a synthetic human virome that allows for the measurement of what is the level of about 206 human viruses that correspond to about 1,000 viral strains. So this is sort of an array-based system based on bacteriophage display, measuring viral components in serological samples from patients. And we need a very small amount of blood to actually measure, less than a microliter. The phage and antibody complexes are IP, PCR, immunoprecipitated, and sequenced. And essentially, at the end, we have a heat map of the patient samples against the viral sequences that are present in their serum. And then we can perform computational analysis to look for similarities or differences in patients who are healthy, patients who are at risk for developing cancer, or patients with liver cancer. In parallel to our VIRSCAN study, we also performed a GWAS study, a genome-wide association study, which allows us to assess the SNPs, or the single nucleotide polymorphisms that are present in the patient. And this gives us an idea of whether there's any genetic association that is associated with the propensity of these individuals to develop liver cancer in the context of a viral exposure. And so this was validated in an additional cohort at a NIH institute, the NIDDK, where they're conducting a prospective study looking at patients over a 20-year period who had chronic hepatitis C, and several of which who have developed liver cancer over that time period. So this was published last year, and we were able to show the host virum infection map for liver cancer. We were able to show that a history of viral exposure can predict liver cancer. A viral exposure signature can identify liver cancer prior to a clinical diagnosis. So it can actually sort of see whether a patient is going to develop liver cancer or not much earlier to the clinical diagnosis. And from the GWAS data, we were able to identify certain genetic variants that link viral exposure to liver cancer. So lastly, I'm going to show an example of a study where we're exploring molecular signatures related to treatment and treatment responses, specifically immunotherapy. And this study is our NCI Clarity, which is our main study from our relatively new now liver cancer program. And so here's just the website for the program in case you're interested in looking a little bit more about what we do. A lot of our science is here, as well as our clinical trials. So that may be of interest. And so this study focuses on immunotherapy and liver cancer. So this is an evolving approach for primary liver cancer treatment. And there are a variety of different approaches within this methodology that are being implemented for liver cancer and other cancer types. However, only a small fraction of patients respond to treatment, and some don't, and some actually experience resistance or relapse. And so at this point in time, it's difficult to determine who actually would benefit from immunotherapy treatment. This is due to the lack of a large comprehensive studies, a lack of a biobank that has resources like biospecimens to interrogate, and a poor understanding of the genomic and genetic factors that contribute to patient response. So as part of the liver cancer program, we began a study called NCI Clarity, which stands for Cancers of the Liver, Accelerating Research of Immunotherapy by a Transdisciplinary Network. This is an intramural cancer moonshot study that's parallel to the cancer moonshots that were initiated by then VP Joe Biden to sort of accelerate cancer research. The aim of this study is to collect biospecimens and clinical data from liver cancer patients undergoing immunotherapy at the NIH Clinical Center and extramural institutions. And again, we're hoping to develop predictors for response or resistance to immunotherapy or acquired resistance to immunotherapy. And so the NCI is conducting this study in concert with a series of institutions and hospitals across the US that are conducting clinical trials where liver cancer patients are being treated with the immunotherapy regimens. We are collecting biospecimens from these individuals and clinical data, housing that at the NIH, and performing correlative downstream studies at the NCI. And so these involve sequencing, so looking at genomics, metabolomics, microbiome studies, radiomics, as well as immune monitoring. And each of these levels will be interrogated separately and integrated. And the idea is to perform computational analysis, again, to stratify patients into homogeneous groups, in this case, responders and non-responders, and to understand what are the signaling mechanisms occurring in each group. and using that information to inform clinical trials and improve patient care. So that's an ongoing study. So here I've shown you sort of three large examples of how we are using this sort of biobank case control study in order to ask questions about liver cancer biology and relating it to clinical management. One where we're looking at molecular subgroups associated with patient demographics. One where we're looking at molecular subgroups in relation to etiology, in this case, viral etiology. And one where we're looking at molecular subgroups related to treatment response. So now we have several cohorts that we have initiated, and that this allows us to do some cross cohort comparisons, so we can compare Mongolian patients with patients who have liver cancer, et cetera. And this helps us to explore many different factors like liver cancer heterogeneity, the underlying etiology and risk factors that are related to this disease, global health disparity occurring within this disease, the underlying molecular mechanisms, as well as efforts to improve the clinical management of patients with liver cancer. And so I'll end there, just acknowledging all of the groups that we're working with these particular studies. This is a large collaborative effort between the NCI and many institutions across the world. And we certainly always want to thank the patients that are involved and contribute to the study. So I'll end there, and I'm happy to take any questions.
My question is, so when you were talking about the Tiger study and how, especially I think it was in the northeastern regions in Thailand, there was like the particular, I'm probably going to butcher this, but the particular like virus or disease that was associated with the fish.
Yeah,
like
cholangiocarcinoma.
Right.
Yeah.
Do we have any idea of like why we saw more prevalence of liver cancer in like the southern regions of the United States? Like, is there a similar kind of regional etiology?
So in the U.S., it's a little bit of a different etiology compared to other parts of the world. So as I mentioned, most of liver cancer occurring across the world is due to hepatitis infection, hepatitis B, hepatitis C. In the United States, the etiology is a bit different. So we see association with alcoholic liver disease and fatty liver disease. And so in regions where there are higher levels of either or, we expect to see a higher level of liver cancer occurring in those regions.
Gotcha. Thank you.
Perfect. Well, with that, we are right at time. Amazing. So thank you again to both of our seminar speakers for being here today. Thank you so much for joining us and giving us insight on your work and on your journey as well, how you became a scientist. Thank you.